Welcome back. In part 1, we explored how important folate is for us and why we need it. Impairments in folate metabolism can include genetic variants such as MTHFR polymorphisms. Individuals with MTHFR gene polymorphisms may have reduced efficiency in converting folate into its active form, leading to elevated homocysteine levels and a higher risk of heart disease, miscarriages, and neural tube defects. There can also be a deficiency of cofactors such as vitamin B12, B6, and B2. These additional B vitamins are necessary in assisting in total folate metabolism and are referred to as cofactors. Clearly, impaired folate metabolism can result in a variety of health issues due to its critical role in DNA synthesis, repair, and methylation, as well as amino acid metabolism. The other important factor related to folate is its proper transport into the cells. This transport of folate is accomplished via several different receptors found in the cells. These different receptors are known as PCFT, proton-coupled folate transporter, RFC, reduced folate carrier, and FRA, folate receptor alpha. The PCFT receptor plays an important role in the absorption and transport of folate, vitamin B9, across cell membranes, particularly in acidic environments. PCFT facilitates the uptake of folate in the small intestine, especially in the duodenum and jejunum, where the pH is lower. It is a proton-coupled transporter, meaning it uses the proton gradient to drive the transport of folate into cells. This receptor is highly expressed in the epithelial cells of the small intestine, where dietary folate is absorbed. It is also present in other tissues, including the choroid plexus of the brain. Mutations in the PCFT gene cause hereditary folate malabsorption, a rare disorder characterized by impaired intestinal absorption of folate. The reduced folate carrier is a membrane protein responsible for transporting folate and its derivatives into cells. It facilitates the uptake of reduced folates, such as 5-methyl-tetrahydrofolate, 5-MTHF, the biologically active form of folate. The RFC is expressed on the plasma membrane of most cells, particularly in tissues that require high folate turnover, such as the bone marrow, liver, and placenta. Defects or reduced activity of the RFC can lead to cellular folate deficiency, resulting in health conditions like megaloblastic anemia, neural tube defects, and impaired DNA repair. Folate receptor alpha, FR-alpha, is a high-affinity folate-binding protein that is responsible for binding and transporting folate into cells via receptor-mediated endocytosis. FR-alpha is particularly important in tissues with a high demand for folate, such as during fetal and brain development. FR-alpha binds to folate and reduced folate derivatives with high affinity, facilitating their cellular uptake. It enables endocytosis of folate by forming a complex with the ligand, which is then internalized into the cell. FR-alpha is highly expressed in certain epithelial tissues, such as the kidneys, lungs, placenta, and choroid plexus. FR-alpha is essential for folate transport during embryonic development, particularly across the placenta and into the developing neural tube. There exist two potential routes of delivery to the brain via the blood-brain barrier, at the cerebral vascular endothelium and the blood-cerebrospinal fluid barrier at the choroid plexus. Folate receptor alpha is neither expressed in brain parenchyma nor is folate receptor protein evidently detected at the blood-brain barrier. However, reduced folate carrier is localized to the vascular blood-brain barrier. In addition, the protein-coupled folate transporter protein is expressed there as well. The choroid plexus represents the pathway for the active transport of substances and metabolites, including folates, into and out of the CSF. It has been demonstrated that the folate receptor alpha plays a critical role in this process and is based on the following observations: that folate-binding protein, folate receptor alpha, is highly expressed in the choroid plexus, and blocking autoantibodies against folate receptors have been found in the blood of patients with cerebral folate deficiency. In this CFD disorder, low levels of CSF folate have consistently been shown even though folate intestinal absorption and folate blood levels are normal. Furthermore, in general, these subjects do not have demonstrable mutations in genes that encode folate receptor alpha, reduced folate carrier, and proton-coupled folate receptor.
Folate receptor alpha is a critical player in cellular folate homeostasis, and its unique high-affinity binding and receptor-mediated endocytosis mechanism make it distinct from other folate transport systems. Its role in development highlights its importance in both health and disease.
Mutations or disruptions in the FOLR1 gene can result in impaired folate transport, increasing the risk of NTDs during fetal development. The other cause of folate impairment, involving folate receptor alpha, will be related to the presence of autoantibodies against FR alpha. In some individuals, autoantibodies against FR alpha can block folate transport, potentially leading to disorders like cerebral folate deficiency syndrome, autism spectrum disorders, and various neuropsychiatric disorders. Folate receptor autoantibodies are antibodies produced by the immune system that specifically target the folate receptor alpha. This receptor plays a key role in the uptake of folate, vitamin B9, into cells, particularly in tissues with high folate requirements, such as the brain, placenta, and developing fetus. When folate receptor autoantibodies are present, they can interfere with the normal function of folate receptors, potentially leading to reduced cellular uptake of folate. This disruption can have various physiological effects, depending on the severity and duration of the interference. There are two main types of folate receptor autoantibodies: blocking antibodies, which prevent folate from binding to the receptor, and binding antibodies, which cause receptor dysfunction and trigger an immune response, further impairing folate transport. Folate receptor autoantibodies are implicated in several neurodevelopmental disorders, such as cerebral folate deficiency, CFD, a condition characterized by low levels of folate in the cerebrospinal fluid despite normal or elevated blood folate levels, and of course, autism spectrum disorders. In fact, several recent studies indicate that a large percentage of children with ASD have folate receptor autoantibodies. Folate receptor autoantibodies have been investigated in relation to conditions like depression, epilepsy, schizophrenia, and other cognitive or behavioral disorders. They have been found in a number of these conditions and, as we mentioned before, especially in autism spectrum disorders. Folate receptor autoantibodies have been investigated in relation to conditions like depression, epilepsy, schizophrenia, and other cognitive or behavioral disorders. They have been found in a number of these conditions and, as we mentioned before, especially in autism spectrum disorders. Again, the premise is that they disrupt folate transport, leading to various neurodevelopmental and neuropsychiatric disorders. A recent focus has been on treatment if one tests positive for folate receptor autoantibodies. Treatment options aim to bypass the disruption caused by folate receptor autoantibodies and ensure adequate folate levels. This includes high-dose folinic acid, also known as leucovorin. This is a form of folate that can enter cells via alternative pathways not dependent on the folate receptor. It uses the reduced folate carrier.
Sometimes, dietary adjustments are also recommended, specifically the elimination of dairy products. Evidence shows that exposure to dairy may exacerbate the levels of folate receptor autoantibodies.
Testing for folate receptor autoantibodies involves a simple blood test known as FRAT, Folate Receptor Autoantibody Test, to detect the presence of these antibodies. This is often done in individuals with unexplained developmental or neurological symptoms or symptoms of autism spectrum disorder. As scientific research continues, it becomes evident that the presence of folate receptor autoantibodies have a negative effect on folate transport. It is advisable to screen for them with the FRAT test, as there may be a possible treatment with the use of folinic acid. As with any medical condition, diagnostic or therapeutic, a physician's guidance is required. If you suspect any neurodevelopment delay, please reach out to your physician as soon as possible.